The overall goal of this procedure is to successfully perform a microinjection into Nasonia vitripennis embryos in order to facilitate genomic manipulation.
This method can help answer key questions related to Nasonia vitripennis biology, such as in venom production and sex determination. The main advantage of using this technique is that it allows for a direct manipulation into the wasp genome and offers a wide variety of advanced genomic modifications. Demonstrating the procedure will be Ming, a postdoc from our laboratory.
To begin, set up several N. vitripennis colonies by placing 200 to 500 adults with a 3-to-1 ratio of females to males in bug dorm cages. Maintain the wasps at 25 plus or minus 1 degrees Celsius with 30% relative humidity and a 12 to 12 light-dark cycle. For 2 days, allow the mated females to feed on fresh S. bullata pupae as well as small droplets of a 1 to 10 volume-to-volume sucrose water solution. For the following 2 days, remove the blowfly pupae to deprive the female wasps of an oviposition site, making them very gravid. To keep S. bullata pupae hosts fresh, store them at 4 degrees Celsius immediately after obtaining them, and only remove them when needed. Differentiate between young hosts that have a red-colored puparium and older hosts that have a darker-colored puparium.